Yes, man. Yeah, man. Yes, man.
Up, Lonesome, up. And that's a meditation, you know. Alright, so hear this now. I have some things, some serious things for going on, but before I do that, right now, I have to, I play some things, but I just play the audio. Yeah. Because certain things have copyright, you don't want to play them. So I play some audio, see? But you have videos, you know, and it's good when people start, you know. You have everything there on the tip of your fingertip right now, you know, where you can just log in and check out and know things. You see me I say? Because hear this now. You have people, them build car, and then we have mechanic, and yeah, mechanic look boy car, and enough man, as the front end the coach, so you find a mechanic. Even if it's a ghetto mechanic, and you trust him. Remember your life that, you know. If him just screw up something good, you know, I can't run away and mash you up, you know, take your life. But you trust him. And you leave the car with him and it come back and it feel good, it a drive, you know, if it breaks start feel or not, but you trust him and, you know, it work. Kind of really want to hurt you. He want to make the money, he want to fix it. Say no want to fix it for you, say in an accident. You understand? But I don't like him couldn't fix it that, you know, accident or something or the other or whatever, you know. So everything come with a risk, see? But, you know, as I said before, me as a man, me just sit down, because you have a lot of people bombard me inbox with some things, and my style that you can bad me inbox with some things where you know me look through that it don't even make no sense, you know. Pastor wanna come say what him a say. Him a scientist. After I'm done preaching, I'm a matter of fact some pastor when them a preach to you and you see man turn to them. Them a preach to you, you know. And you see man turn to the pastor them and you see what them a preach to you and you ask them some question, them vex. Because them don't expect you to ask them the question either, you know. You understand? They don't expect it. No, they don't expect it. So you want me to know? Boy, I know about vaccine and them things. I tell you something, I watch them things, you know, cross meeting and things. And not like somebody will come tell me, boom, bam, bam. The injection need where them use and juke you and the thing them where them, like how that tell do so boom and put a stint in the yard, but you have to run. They might run a cut your risk your grind and then run the thing in your through your vein and look at your veins, they run it through it. And it go through it. I'm running your heart and put the stent in it and cut you and them thing there. You know say Jano. Them thing Jano. You know, it work away, don't it? And them thing enough man saved by them thing they set speed, artery black up and all kind of things and all you see? So you have enough man where them do a lot of study on it, you know. So me want to know what is antibiotic and them want to know what's the vaccine and what build it. So me just play some things because we have it in a visual to what I play them, you know. See, let me see. Antibiotic first. Play what is antibiotic. And know who first come up with antibiotic and how it help much life. You know, blessed. Hear this. This is what me listen to, you know.
Antibiotic in 1928. Unknown to him at the time, his findings would quickly revolutionize the whole field of medicine forever.
So, I could come back and say who invented antibiotic.
Alexander Fleming discovered the first antibiotic in 1928. Unknown to him at the time, his findings would quickly revolutionize the whole field of medicine forever. So we ask, what is an antibiotic and how does it work?
Yeah. Remember like ask the question them, you know? Watch them and see how them work and them things.
Antibiotics are compounds that kill or inhibit the growth of bacteria. They do this in a variety of different ways. Penicillin works by breaking down bacterial cell walls, killing bacteria and preventing further growth. Imagine it like popping a party balloon. Since the discovery of penicillin, many other antibiotics have been found and modified to make them more effective, as well as ever increasingly safe for human use. Different antibiotics work in different ways to kill or stop the growth of bacteria. Antibiotics like erythromycin and neomycin work by stopping protein synthesis inside the bacterial cells. By binding to specific RNA and other internal structures, these antibiotics stop the process of protein synthesis, which is a basic need for all living organisms to survive. Antibiotics like ciprofloxacin, on the other hand, bind to DNA enzymes, inhibiting DNA replication, which stops cell mitosis so the bacteria cannot divide and grow. However, in some cases, bacteria can become resistant to certain antibiotics. This happens because of natural selection. Some bacteria can contain DNA, which allows them to make enzymes capable of breaking down antibiotics. These bacteria will survive such pressures, allowing them to proliferate and share their DNA with other bacteria, effectively giving them the same ability. Viruses are naturally immune to all antibiotics. This is because they do not perform the biological processes that bacteria do in order to survive. They also don't have cell walls. To make it easier and slightly more fun, imagine we give your mother-in-law a sledgehammer. We've also taken your car. Sorry about that. Luckily for you, we give you a tranquilizer dart. You should find it quite easy to prevent any damage being done to your car. However, the second time round, we remote control a small drone to fly into your car through its sunroof, then have it connect via Bluetooth to transfer software to disable your car's brakes. A little harder to stop with a small tranquilizer dart, don't you agree? A virus isn't a living organism, and will only do damage to a cell once inside, whereas a bacteria will do damage to a cell from the outside and can be killed, or at least in your mother-in-law's case, put to sleep for a little while. Antibiotic resistance in bacteria is becoming one of society's greatest and most frightening problems, facing us with a huge challenge over the coming decades. Many antibiotics are found in nature, but as we continue to destroy our natural surroundings, such as the rainforest and marine environments, where else do we look?
So many antibiotics found in nature too, but like I'm saying, them destroy the rainforest. Because you have something we use as antibiotic, a natural antibiotic, Yeah, man, and some remedy we have and thing, but you remember them destroy enough of them things themselves, So we continue antibiotic and never go up and do that thing with them at all, go to them and we run up and down with me. I have time for that. I want the reverend come chat to me and them thing, man. I think them different,
In this video, we're going to talk about the mode of action of antibiotics. So we're not going to look into extreme detail, but we'll just look at the overview in a way. So mode of action of antibiotics. Now we have to know that antibiotics either destroy or slow down the growth of bacteria.
So people have sent me things, them have to break it down like this. Come watch it, I watch it. Me just play the audio, I couldn't hear it, see? I'm sending you some link when you can watch and them thing. You don't have to do no one research, see? You want to know what is antibiotic and you want to see and everything about them thing. Everything fascinated them have something about it. It's there. Not for a man to interpret what he want to interpret. And then think upon you and tell you, so boy, them want to do this. Me know that. No guy. Round here, none. See? Certain things fascinate me, Someone know about it. Yeah. I understand? Some study about it. Go and play.
So therefore, if it destroys a bacteria, it is bactericidal. If it slows down the growth of bacteria, it's bacteriostatic.
Okay.
Here I am drawing a typical bacteria with cell membrane and cell wall. The cell wall and cell membrane structure I am drawing is characteristic of a gram-positive bacteria. But we will just assume it is any type of bacteria. Within the cytoplasm, within the bacteria, we have a circular DNA. We can have RNA being synthesized and ribosomes here. Ribosomes will synthesize proteins. The first type of antibiotic are the ones that I want to talk about, are the ones that target the cell membrane. These antibiotics will disrupt the cell membrane function, which means that they disrupt the phospholipid bilayer. So here is the cell membrane, and here is the cell wall, known as a peptidoglycan. They are different. The antibiotic binds here to the cell membrane. It will alter the cell membrane structure and will make it more permeable. This will just disrupt the osmotic balance, causing leakage of cellular molecules, and essentially it will increase the water uptake, leading to cell death. Examples of these drugs are polymyxins, which I spelled wrong, and polines. Now, polines are actually specifically used against fungal pathogens.
Yeah, man. Same
But
time, yeah.
that's just a note to know that you can use polines that target cell membranes.
Micah, no to you. So when I use that, so you're some more like a learning channel. See, when I use anti-vax and them thing, they know to you. You get banned right away.
In fungus. So polymyxins and polines target cell membranes. Other antibiotic classes target the cell wall. These antibiotics will inhibit cell wall synthesis, so these drugs are therefore bactericidal. They will cause bacteria to die. Example of these drugs are penicillin, cephalosporin, and bacitracin. I hope I pronounced that right. All these drugs have different mechanisms of action when it comes to inhibiting the cell wall to synthesis. A good thing about these drugs is that it won't affect human cells because human cells do not have peptidoglycans. We do not have cell walls.
Okay.
Penicillin and cephalosporin are beta-lactams. And then you have the bacitracin and other glycopeptides that prevent the synthesis of cell walls as well. Antibiotics that disrupt the cell wall is pretty extensive, and so hopefully I will make a video on that. Cell walls are important to bacteria. Disrupting the cell wall or preventing in cell wall synthesis will result in cell death.
All right, so me just cut again. If somebody explain something to me, come me have the visual. You understand? Me just play the audio, so me just go and link to that. Somebody explain something to me and say, hey, auntie this, that and that. You have to come break it down, tell me what protein, everything, and show me and why it's bad for you and all this make and thing. You can't do it. They listen to the specialist. You don't go come and just follow because some guy make money off of these things and follow and say boom 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 and sell. Say my antivax should not sell some things to make money off of it. You understand? Me don't want you from one. Nah. So you have to explain and break down the thing to me and tell me why this not good and why it good and why it can work. You can't do that. Move! You understand? It don't make no sense. And people want to start thinking for themselves. That's why you see enough people rather go up on a crate and buck up more than listen to the truth and research the truth. Me don't have foolishness with none of them, you know. Be different, man.
The next type of antibiotic class is the ones that can inhibit here DNA synthesis, or what's occurring here, RNA synthesis. So these antibiotics will inhibit RNA and DNA synthesis. So here the bacteria are typically having DNA replicated, of course, like so. So we have antibiotics that prevent from DNA from being synthesized. These are quinolines and also a class known as nalidixic acids.
So when each time I'm talking, you don't know your cross-reference and thing there, you see the video and then you just look up quinolines, what the meaning and where to, and you look up where the acid is, the vital role where it play and anything. That's your cross-reference thing then. You know, you just can't single them out and cross-reference a thing. And they must show you in a visual, you know. See? And you have name and everything, antibiotic, you see, and your search work and everything, and you know, the side effect and everything. So me like, you know, my thing, them have to just come to me like this. No guy can just build something and say, yes, they want to do this, like the 5G. Remember that? At the same, because dirty thinking people, them come with 5G last year, and you notice how them just fade it out and just step on no vax, anti-vax. No, them that come with 5G until they said that this thing not real. And the 5G had killed people. You just remember good enough. You understand? No, they fade it out neatly. Nobody now talk about it again. You see? And 5G, what? 5 generation. I'm going like 25 times faster than the 4G. So you can download and use your phone and video with it and broadcast with it and even better, you know, better quality and everything. And them come with them bag of thing with it. It a drop down bird and thing. Them same one. I know them come with something else. You not listen to them. Them a try to make you destroy yourself. You understand? Look about the thing, man, and research your thing. That mean a guy can go in your head, yeah? You understand? Yeah, man.
And then, of course, RNA can be made from the DNA. And there are drugs such as rifamycin, which prevents RNA synthesis and therefore protein synthesis.
So you say
other classes of antibiotics also inhibit protein synthesis itself. Protein synthesis is carried out typically by ribosomes. which translate mRNA into proteins. Bacteria need to make proteins in order to survive. However, there are antibiotics that target either the 30S subunit or the 50S subunit of the ribosomes. Erythromycin and chloramphenicol target the 50S subunit of ribosomes, whereas there are tetracycline, streptomycin, and gentamicin that target the 30S subunit. Either way, this will disrupt the ribosomes from making proteins, which will result in the bacteria obviously unable to make proteins, so the bacteria is pretty much useless. Thus, the bacteria can still be alive, but it just is unable to make anything. And so these antibiotics that target protein synthesis are bacteriostatic. They prevent bacterial growth. Last type of antibiotic are the ones that inhibit folic acid metabolism. So normally, this green thing here, called PABA, which stands for para-aminobenzoic acid, it's a precursor to folate. So the bacteria can metabolize PABA into folate. There are antibiotics out there that inhibit folate or folic acid metabolism. See, PABA normally can be metabolized to folic acid within the bacteria. And folic acid, or folate, is essential for the synthesis of adenine and thymine, 2 of the 4 nucleic acids that make up DNA. These antibiotics, such as sulfonamides and trimethoprim, prevent conversion of PABA to folate and therefore prevent proper DNA synthesis, you can say. Humans do not synthesize folic acid, and so these antibiotics are okay to take for humans and are selective towards bacteria. I hope you enjoyed this short video on antibiotics.
All right. So these are the things, when you explain something to somebody, you know, you have the visual and there and then work it out and show you how you just look at how them create these things. You know, you cross-reference a thing them and you have it there. The vaccine now, go to the vaccine now. Like how some people that say them don't want the Johnson because, you know, the other one look like it hold up the approval rate more. Alright, but I understand more about the same vaccine now where you see them around. By the way, me see the star come out just now, before I play that. Me show you the star come out. In Jamaica and everything, I hustler make money, you know. Yeah, man, make money, man. Serious money to man. See the star does come out now. Look where them start though. Yesterday, in every place across the world, you know, them that give somebody something for, you know. Yeah, man, bless up, Dixon. They might give somebody something for, you know, for, because they want done this pandemic, right? In Jamaica now, them come up with something with the ministry, you get nothing free, and you give it to some people. I say, okay, I can give them as an MPA. Must $1,000 for get a joke, because you skip the line. Why making the line in the first place? Now, see it now, private health facilities, and the private health, no, you know, facilities in Western Jamaica require a deposit of $2 million for each COVID patient, because me see the guy from one of the university in Manchester, down a Mandeville, him drop out just now. See? So I know those poor people that grab up, so in the poor places that grab up them because of them they run out and them want to do our set to grab them up. So them have think some money. Look, see that? Look here. You can't believe this? When them talk one thing and them say one, you know. See that? The people. Look here. That are the headline that's two hours ago. You see it? Private, you see what them do? Eh? When one come and say boop bop and then them turn around now. And it's so much money them want for it. 2 million to admit. And then you have some people now that mean they may have oxygen and give a little better bed than them thing, then them want 2 million. Poor people can't pay this. So when some man I tell you, say, yeah man, not take nothing, remember, that out of your reach, by the way. That out of your reach. You know, have no bed for go up on them thing there. So them are half the bed and give you condition to them private thing. This is how Jamaica when it's a bandula place. You understand? It's a bandula place. Them dangerous. See? And that out a poor man reach, because you never 2 million, just say admit and then you don't know. Saying you say, okay, I better me go there so. That after the man them have the money. And they might get a better treatment and thing. You don't have the money now, them just fling you. Yes, at 31 a day. 17, 18 a dead one time. How much a dead one time? Eh? You understand? It's a goddamn shame. See? But as I say, anything at all, where the eye them, you know, a man can just send things and just say boom bam to me because me want to see where they, how the thing them, how them come arrive at certain things and how things made and how things work and how it operate. Me want to know that. You understand? Because if on the earth you want to know that. Me don't want no guy come tell me what they want to tell me. Me know that. You understand? So some people now, even at the stage with the Johnson say, oh yeah, different rate because that show you 95%. But make it break down. As me say, we have the visual, but me not gonna show the visual because you know say. You understand? So but we go into the link to it. Just hear how it don't make no certain difference. I do the one dose. Just listen.
This is the new one-dose COVID-19 vaccine from Johnson & Johnson. In early March, more than 6,000 doses were supposed to be shipped to the city of Detroit, Michigan.
Listen.
But the mayor said no thanks.
Moderna and Pfizer are the best. And I am going to do everything I can to make sure the residents of the city of Detroit get the best.
So in the things I does the best because you show the 98% kind of rate. So it can tell you everything where it do and how it work and operate. So I want to understand. I want to watch these things. Don't make no man come telling you nothing. Them are not dark, them not even for flushing a toilet. Toilet too good for some of them. See? No make them telling you nothing, man. All when thing reaching at your house and when you look at your own family have the thing. And I struggle with the thing, them still are coming and push them little dirty self. Them same one to the 5G. Them same one. Them bombard my phone. I'm ever tell them my foolishness. Me no yes man. We don't study about the first generation, second, third. And when you look now, at the same phone them what we are using, you know, just better technology and it improve. And them are born it. Till our youth get serious and are burn down cell towers. Them show bird that drops at the 5G and do it and that fry your brain. And this thing not real. It's a pandemic, so it's not real. Yes, still I have one family now a drop. Look how much people drop in Jamaica, not figure. Are real people them? You see? You know, if you have a real crisis where you don't still listen to them little nastiness here, you don't have the money. And make sure when you go on like you eat that you know pan the floor where them put people and you don't have no oxygen to not catch your breath. Make sure you're alright. You understand?
He was referring to these numbers, the vaccine's efficacy rates. The vaccines from Pfizer, BioNTech, and Moderna have super high efficacy rates, 95 and 94%.
Yeah.
But Johnson & Johnson, just 66.
66.
And if you only look at these numbers, it's natural to think that these vaccines are worse than these. But that assumption is wrong. These numbers are arguably not even the most important measure of how effective these vaccines are. To understand what is, you first have to understand what vaccines are even supposed to do. A vaccine's efficacy rate is calculated in large clinical trials, when the vaccine is tested on tens of thousands of people. Those people are broken into two groups. Half get the vaccine and half get a placebo. Then they're sent out to live their lives while scientists monitor whether or not they...
So you get tens of thousands, send out half a placebo and half with the vaccine, and then them do it. But what Johnson do it, send out a bigger, because them do it in the US, but Johnson send like South Africa with this strain and other places. So you see them get 64, they kind of rate what them talk about. That no make it no good. So them are going to break down and tell you what really happened. Just listen good.
Get COVID-19 over several months.
Over 7 months.
In the trial for Pfizer BioNTech, for example, there were 43,000 participants.
43,000.
In the end, 170 people were infected with COVID-19. And how those people fall into each of these groups determines a vaccine's efficacy. If the 170 were evenly split, that would mean you're just as likely to get sick with the vaccine as without it. So it would have a 0% efficacy.
Okay.
If all 170 were in the placebo group and 0 people who got the vaccine were sick, the vaccine would have an efficacy of 100%.
Okay.
With this particular trial, there were 162 in the placebo group and just 8 in the vaccine group.
Okay.
It means
so it means 8 in the vaccine group, it gets sick, and the 100 add over the placebo group. So it gives you the percentage there, so the 98%. You know,
those who had the vaccine were 95% less likely to get COVID-19. The vaccine had a 95% efficacy.
Okay.
Now, this doesn't mean if 100 people are vaccinated, 5 of them will get sick. Instead, that 95% number applies to the individual.
Okay.
So each vaccinated person is 95% less likely than a person without a vaccine to get sick each time they're exposed to COVID-19.
So that means not like you won't get sick, but just look, just the and this and then that. So it's a 95% you less than a man when I get it, okay?
And every vaccine's efficacy rate is calculated in the same way.
Calculated.
But each vaccine's trial might be done in very different circumstances.
So one of the biggest considerations here when we look at these numbers is the timing in which these clinical trials were performed.
This is the number of daily COVID-19 cases in the US since the pandemic began. The Moderna trial was done completely in the US, here in the summer.
Okay.
The Pfizer-BioNTech trial was primarily based in the US too, and at the same time.
Same time.
Johnson & Johnson, however, held their US trial at this time, when there were more opportunities for participants to be exposed to infections. And most of their trial took place in other countries, primarily South Africa and Brazil. And in these other countries, not only were case rates high, but the virus itself was different. The trials took place as variants of COVID-19 emerged and became dominant infections in these countries, variants that are more likely to get participants sick. In South Africa, most of the cases in the Johnson & Johnson trial were that of the variant, not the original strain that was in the US over the summer. And despite that, it still significantly reduced infections.
Yeah.
If you're trying to make one-to-one, head-to-head comparisons between vaccines, they need to have been studied in the same trial with the same inclusion criteria in the same parts of the world at the same time.
If we were to take Pfizer and Moderna's vaccine and redo their clinical trial at the same time that we saw the J&J's clinical trial, we might see quite different efficacy numbers for those.
Yeah.
These efficacy numbers really just tell you what happened in each vaccine's trial, not exactly what will happen in the real world. But many experts argue this isn't even the best number to judge a vaccine by anyway, because preventing any infection at all is not always the point of a vaccine.
The goal of a vaccine program for COVID-19 is not necessarily to get to COVID-0, but it's to tame this virus, to defang it, to remove its ability to cause serious disease, hospitalization, and death.
It helps to look at the different outcomes of an exposure to COVID-19 like this. The best case scenario is you don't get sick at all. The worst case is death. In between, there's being hospitalized, severe to moderate symptoms, or having no symptoms at all. In the absolute best circumstances, vaccines give you protection all the way to here. But realistically, that isn't the main objective of COVID-19 vaccines. The real purpose is to give your body enough protection to cover these possibilities. So if you do get an infection, it feels more like a cold than something you'd be hospitalized for.
Okay.
And this is one thing that every one of these COVID-19 vaccines do well.
Okay.
In all these trials, while some people in the placebo groups were hospitalized or even died from COVID-19, not one fully vaccinated person in any of these trials was hospitalized or died from COVID-19.
One thing that I wish that Mayor would have understood was that all 3 vaccines have essentially 100% effectiveness in protecting from death.
The mayor of Detroit did backtrack and said he'd start taking Johnson & Johnson doses because it's still highly effective against what we care about most. Efficacy matters, but it doesn't matter the most. The question isn't which vaccine will protect you from any COVID infection, but which one will keep you alive or out of the hospital. Which one will help end the pandemic? And that's any of them.
The best vaccine right now for you is the one that you're offered.
With each shot that goes in someone's arm, we get closer to the end of this pandemic.
So that me want know, you know, when you watch a video, let's say everything, you want to know how them develop up, you want to know these things, them there. Them is there. A car, a truck, a bus, everything, gun. You notice now, them now come and burn the bullet or the bullet make a nothing at all, and you still take life like crazy. But something if it's do some boom boom and so because in a pandemic, everybody just jump on it. Not everybody, you know, them but want spread what them are spread. And look where it starts from, them just jump on it. Them jump on the 5G, when you get 5G the other day. Them jump on it to me, yeah man, it a kill. And man come tell me, yeah man, it a kill, yeah man, we are talking about the fry your brains and it a mash them a kill it, we all away. It don't make no sense. So if you have a country where the #1 country in the world, if you just sit down and just think, right? I have a rising power like a China. You think a country like US where you're going to everybody country and do all kind of thing, man get up and kill them people and then make a China run in pan them and start take them soldiers and take them themselves and take them over. Not like that. And if you kill your people them, kill half your people them, then you're weakening up yourself. See? So even these things don't match. When you think about the reality of life. Yeah, them want to kill 2 billion away. Simple by your drinking water and simple by your food. You understand? That simple just keep silent, you just a drop and you never even know where it reach you. You know, biological agents and all kind of things. See? And them have it. Them have it in the lab. We know that. You understand? So we know them can deal with biological warfare and all kind of things. But this try you for how and why. That will not build them nation. And yes, still them build it, you know, and yes, still them, but them do them and give them people first before third world country, some place where we start get nothing yet. You still them want to kill me, but I just tell them they will get something, we have to sit down and wait. So if them that build it and then they'll send it by you first and send our people third world and just give Africa and give and go to third world first and sit down and follow them and watch the what way I get look, man. Them no give us a man. Them a look about themselves. So these are the things, you know. You have to watch too, you know, man, I look about themselves. They want to look out for themselves first. All of these countries look out for themselves first before them even turn to you. And you say, okay, you question these things. But enough man don't want that. Them just want to hear. Because people listen lie than the truth, you know. And people listen stories. A man just come up with something. And one woman have face swell. And she said, why me take a shot? You never see she take a shot. She don't prove to you. She take no shot or nothing. She just come and have maybe some loop. She have some problem. You understand? Live not problem or something and she just come do something and her face been up her cancer or something. And you say, yes, I do it. Because the woman, yeah, man, you never see her. They really believe in them thing there. Them just show you without evidence or something and you run with that. Eh? Nah, man, you can't make your brains a shallow, man. Me not know that with a guy, man. You see? Let's remember just want to hear about this again. Yes,
how do you create a vaccine to fight a new disease? And why does it take so long? Well, first, you have to identify the virus or bacterium that causes the disease. In some cases, the pathogen itself can then be weakened or deactivated and used to make a vaccine.
So them that use the same germs and make the vaccine. Yeah? Yeah, man. So them that use the same germs and make the vaccine, break it down, the pathogen. Yeah, them just listen how them that make it and hear them derive a certain things.
But this isn't always possible. In these cases, the next step is to identify the pathogen's antigens, unique protein or glycoprotein markers, which can form the basis of a new vaccine. For some pathogens, these antigens always stay the same. Other pathogens, like the virus that causes flu, can change or mutate their antigens, and so these must be reanalyzed every year. Once you have identified the pathogen or antigens, you need to decide how you are going to use them to build a vaccine. This is called the vaccine platform. There are a lot of options here, like subunit vaccines that only use part of the pathogen. There are also newer or more experimental technologies, like viral vector or nucleic acid-based vaccines. But we'll get to these a bit later. You also need to decide how the vaccine will be administered. By injection into the skin or muscle, orally like the polio vaccine, or through the nose like some flu vaccines. Each of these options could affect the response the vaccine induces. For now, let's say you've decided on the platform.
I don't know where you read from, but you have to go on to. Because we tell us so we not in that we do not say there is no evidence when me I look on whole evidence, me I look how the thing make and them thing. You know see you somebody get something, you ask them a doctor and thing, you check out them background, ma'am. Yeah, said there is no evidence to do what? Eh? When come out for the 5G now and then go and just live for the vaccine now, when they believe in the 5G territory, you know. So the whole want to come out for that now, where we depend that now and depend something else. Eh? I lose people, I lose them loved one. Especially in Jamaica, 1,500 gone already. See, we don't want to hear from money. We don't want to hear from money, man. We'll start banning the street.
And designed the vaccine.
Yeah, man.
Next, it needs to be tested.
Yeah, man.
First in cell cultures and then in animal models. This might include challenge studies, where animals that have been given the vaccine are deliberately exposed to the target disease to see if the vaccine protects them. Further down the line, and if the disease isn't too serious or effective treatments are available, humans might take part in challenge studies too. Once the vaccine candidate is proven safe in animals, you need to prove you can manufacture it to a high enough standard for people and at scale. This is called GMP certification.
GMP certification. So this I explain, I take you right through the whole vaccine and how them come by it and how much years and them can't skip nothing but them can cut it down in a 12 to 10 to 12 months or to 14 months. But them can't skip nothing. Them have to go through the same process. And the reason why it take 7 or 10 years are because when them send it, them watch the whole world and put you out there. Just listen and it will tell you. See? And what them are tell me. Them not even janitor hospital. Me not listen to them. Because nobody know want to clean them hospital toilet. So I listen to them. So them are not janitor. You know, I go up to no porter much as reach nurse, much as a doctor, not much as a professor or scientist. What may I listen to one of her? After me know that.
If the vaccine passes all of these stages, it's time to begin human trials. Normally, these come in 3 phases, and they all have to be passed.
And they have to be
First, the vaccine is given to a small group of healthy people to test for adverse reactions. This is called the safety trial.
Safety trial, okay.
Second, you give the vaccine to hundreds of people to work out what dose is needed to trigger a big enough immune response.
Okay.
Finally, you can trial the vaccine in thousands of people to see how effective it is.
Okay.
This step is often the slowest, and participants are sometimes recruited around the world.
Yeah.
Researchers have to wait for participants to come into contact with the pathogen naturally.
So them wait, so the big trial, how them do like first, second and third stage. So them send it like around the world. So them want everybody and watch it when you come to the, you know, pick up the thing naturally and watch it, so it take time. So them want to see how it operate. Instead of infect you, and them send you out and maybe that place have some, you know, some of the disease and things and see when you run through it, or it affect you naturally. See? So it take time. Just listening.
By tracking how many become infected, they can work out how effective the vaccine is at protecting the group.
Okay.
It can take decades to gather enough data to be sure.
Yeah. That's why them take the decades. And I like, and for make it take decades. How them send it out and the researchers sit down and them wait and them want to get a like run worldwide. But you have emergency thing when you have pandemic. Where you can't skip none of this face them, but you can't shorten up. Just listen.
But if you do, the vaccine can finally be licensed.
Yes, okay.
There are still problems to overcome, however. Mass producing a vaccine to a high standard can be a difficult job. And once you've cracked production, you still have to deliver the vaccine to where it's needed. And then there's phase 4 monitoring, keeping track of rare adverse reactions that the vaccine might cause, even though it's passed the previous trials. This trial process is very safe, but that comes at a cost. It is also very slow.
Very slow.
In an emergency, there are ways to speed things up,
Okay.
like reducing the time spent waiting for paperwork to be completed.
So they wait for paperwork and everything to complete it because them, when they put out the study, them wait for years and see how it operates right across. See? So then just listen and you know why the thing, man, not say the vaccine and bill. The amount of thing them push it through and inoculate the people them out there and wait for it naturally for, you know, and then them can know, say, okay, this are the dose, that are this, alright. them sickness are, it work that way there, and them gather the whole of these information. So it take years.
We're running different trial phases at the same time.
You're running different trial
but
phases at the
steps
same time.
are never missed out.
Yeah,
By
alright.
working
So me just rewind back to them. So now step never miss out. So although you run it at the same time, no step can't miss out same way. When you have pandemic.
emergency, there are ways to speed things up.
So when you have emergency, like when you have a pandemic. Yeah, man, rewind on. So me like understand the thing, and I don't want them explain nothing to me, and I want the scientists, I want the people them how the thing make and make it. You understand? So emergency, you know, them tell you how it speed up.
Actions that the vaccine might cause, even though it's past the previous trials. This trial process is very safe, but that comes at a cost. It is also very slow.
Yeah.
In an emergency, there are ways to speed things up,
Yeah.
like reducing the time spent waiting for paperwork to be completed, or running different trial phases at the same time.
Different child phases at
But
the same time.
steps are never missed out.
Our step
By
never.
working more efficiently, a protected vaccine could be produced in as little as 12 to 18 months.
12 to
And
18 months.
advances in biotechnology could speed up production pipelines as well. And there are more ways we could speed up production. Let's come back to some of those newer vaccine platforms.
New vaccine.
Nucleic acid vaccines don't use pathogens or antigens directly.
Yeah.
Instead, they deliver the genetic templates for the antigens. These DNA or RNA templates can be delivered directly or in lipid nanoparticles that help them to enter cells and improve their stability after injection. These nanoscale fat droplets encapsulate the RNA or DNA, protecting it as it enters the body. Some lipid nanoparticles can also act as adjuvants, molecular triggers to kickstart the immune response. Once
So
inside the body,
kickstart your immune response. Because your immune them are used now for kickstart and fight. Your body really fight the thing off, you know. Yeah, man. All the people then say them need more job because them think and I screw up more, you know. See? Just listen.
these DNA or RNA templates instruct our cells to start making antigens which activate the immune response needed to build immunity.
See it?
RNA and DNA are much easier and faster to produce in the lab than antigens are. They're also potentially safer than vaccines that contain whole pathogens.
So the vaccine first time used to contain the whole pathogen, the whole germs, and you catch it and you write tonight. So they want to know and your whole immune system, it make it speed it up and recognize it quicker and go deal with it. Instead of that. That's what they just say now. And they like, you know what is this? Them know what it is, them ever study, them ever watch it, them ever watch a scientist or watch anything, them ever watch what them I give them. You understand? So they could develop people.
Which activate the immune response needed to build immunity.
Yeah, man.
RNA and DNA are much easier and faster to produce in the lab than antigens are.
Yeah.
They're also potentially safer than vaccines that contain whole pathogens. That means they could hopefully be used by people with weakened immune systems.
See it?
But nucleic acid vaccines are still experimental. Another approach is called a viral vector.
Viral vector.
Viral vector vaccines work by inserting an incomplete segment of genetic material from a pathogen inside a harmless virus that doesn't cause disease.
So
This then acts as a vehicle for the genetic material.
That's what them not use from long time. Viral vector. Where them use the viral. Yeah, just listen to viral vector. So this is the one with them that use before.
Delivering it to the right place in the body where it can be translated into proteins, triggering the immune system. The viral vector may even be self-replicating, increasing the amount of vaccine in the body. Viral vector vaccines are also quick to produce. Safe vectors have already been established, and it is relatively easy to insert different target genetic material into these established carriers. And as the genetic material inserted is incomplete, they can't replicate and cause disease, so they're also very safe. But there is only so much the process can be.
Me here. Cannot call me here. Can't do that. Can't do that, man. can't call me from faith. Let me just talk to the woman quick, yeah? And people just, Jano, can't just call me, say, you know. Can't just call me, say, man. Yeah, man. Yeah, man. I do my thing, man. I have to do my thing, you know. I have to play my part, you know. You think we sit down there and say, oh, we don't do this and that, and they still, when we look at our loved ones, them are dropping, and them drop. More than even gunshot and everything for this year. See? And then you have some people who want to come and tell us, say, blah, 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 no, we don't know what put. Yo, nobody come tell me nothing. You understand? Because none of them, and I like them send this to you, or them sit down and watch these things, you know. Them have some little devil people who go on and send something to them and say, no, you know this. And you don't know the meaning of this. You don't know where, you don't know nothing about your body. You don't study nothing at all. See? Nothing at all when me attack you. So them now sit down and study it and study about antibiotics and about vaccine and how it works. And you know what is this? And they try to kill you. And I bring that to me, man. And your own body will fight the thing, you know. You understand? And then they'll kill the virus or them that kill and then use it and then put code in that and right and then them joke with that. No. And you see me I say? I like a little branch off of the thing so they want to use the whole thing with them have and deal with like back in the days. You know?
How do you create a vaccine to fight a new disease?
So let me run through that one, you know. The response on a vaccine.
Vaccines and the immune response. How vaccines work. Influenza vaccines are able to trigger an immune response by mimicking viral infection. They are usually manufactured using inactivated or killed virus particles taken from various circulating influenza strains.
Yeah, kill virus.
Inactive fractioned viral components contain the subvirion particles hemagglutinin, HA, and neuraminidase, NA. In Canada, influenza vaccines are administered via intramuscular injection. These fractioned particles containing the foreign antigens hemagglutinin and neuraminidase are released from the vaccine into the bloodstream. There, they are met with an immune response mediated by various immune cells, including macrophages, T lymphocytes or T cells, and B lymphocytes or B cells.
So B lymphocytes and T lymphocytes, T cell and B cell. So these are things that when I can go look up, oh, I never hear about B cell and T cell. Just go look them up and I will hear the name and everything I know. As someone when they talk to me, they have to talk to me, you know. Yeah, sense in the doctor way, you know. No foolishness come to me, boo, 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 boo. I'm around with that, you're mad.
A major mechanism of action involves macrophage phagocytosis of hemagglutinin, breaking it into smaller components. After ingestion, macrophages display the hemagglutinin antigen on their surface in combination with a specific receptor known as the major histocompatibility complex, or MHC. T cells are now able to recognize and bind foreign antigens that are associated with the MHC. Upon binding to the MHC receptor, T cells become activated and proliferate into either cytotoxic T cells, regulatory suppressor T cells, or helper T cells. Activated helper T cells express hemagglutinin receptors specific to the vaccine strand on their surface and play a major role in antibody generation and memory B cell activation. Unlike T cells, B cells are able to ingest hemagglutinin independent of the MHC. Once internalized, B cells process the hemagglutinin antigen and present it on their surface in combination with an MHC. When activated helper T cells interact with activated B cells expressing antigen MHC receptors, they begin secreting lymphokines, which have several effects. Lymphokines trigger activated B cell proliferation, which leads to either their differentiation into memory B cells or into plasma cells. Plasma cells produce hemagglutinin antibodies specific to the strain of influenza contained in the vaccine. Memory B cells aid in future immune response when exposed to an active influenza virus. When an infected host sneezes towards an uninfected person, the nasopharynx is exposed to aerosol droplets containing whole, live influenza virus.
Once inhaled, the influenza virus attempts viral colonization of nasopharyngeal epithelial cells. Assuming that the vaccine strain matches that of the live virus, hemagglutinin antibodies block viral attachment of live influenza virus to host epithelial cells, and overall disease is either avoided or diminished in severity.
Okay. Some of that, listen on. Yeah. If you have a new, but you know you can't prepare for your funeral either if you just drive out. So why me wanna tell somebody say okay, and then them live on something and then leave people down the path and do them say people are dead. And so we weak immune system. But you know make sense. You can't say if you have a weak immune system, and you don't know this and that and get the world sweat skin, then you see more people are dead, And you contribute to that. You understand? By taking people, because people just take things and just say, all right, it's scary, Yeah, but you're going on a plane where we never even know the man who have flight, and when I know like him, just holla and drop the plane with you. $300 you one time, and you drive a car when I know I come crush you. You have to do something. You have to.
This is how coronavirus invades your body, sinking its crown-like spikes into your cells, using molecular deception to pick their locks, and hijacking your body. But there is one way to prevent this, by using one of the virus's weapons on itself. Hey smart people, Joe here. Right now, there's about 7 billion people all waiting for the same thing. A vaccine that will protect us from the virus causing COVID-19. And if you're like me, you want to know what's in it, what makes it work.
Yeah.
Making a vaccine and getting it out to the public is a long process with a lot of steps, so we can make sure the vaccine is safe and effective.
Yeah.
And it's pretty typical for that to take 10 years or more. Now, in an emergency, like this pandemic, well, we can't skip any of those steps, but we can speed this up by doing some of them at the same time. But none of that can happen.
So your class reference, you can't skip none of the step. But you can't do the reference to speed up something because through that and that. And another first thing you have swine flu, you have this, you have the Spanish flu, we kill over 50 million people. That's why I say I don't want to hear from some of them people because me is a man, but you have the black death, the plague. You have all kind of things that corona and all kind of germs and things that kill people a lot before. You understand? You have all kind of things that reach people, and because at these medicine and people work on some things, and you build up certain things that come with some of the things. I save the world. And then you have some people now come in 2021 and want to come tell me something. But yesterday if you crash, I'll get gunshot to go in there. And the same company they make where you listen. Enough people do have plastic surgery right now, Let me tell you this. When you see doctor push all them things and you move them things, and a C-section. If them not give something for thin out them blood and them thing there, let me tell you this, that blood clot that kill you a long time. You never even come off of the table. Them so advanced that them are cut you and them are put you to sleep. See? And you get up out tight and you come out tight and you're good. See? And then they make one big thing over something, and although people are dead, you have some people come and attack them bag of crap. Me no unite with none of them. None. You understand? You drive out, a big truck can ride off a car right now. None of them. See? Have a fighting chance. Me not listen to them.
Until you figure out the first step. What do you put in your vaccine that will make it protect people? And that's what we're going to talk about today. We're going to visit a lab and meet some scientists who study the coronavirus. We're also going to meet a really big, awesome science machine and learn how they used it to design this, the key ingredient inside the very first COVID-19 vaccines.
But even if it's a specific one, in a 1928, at the first them I come with the antibiotic, the penicillin. First time you must have a first. You know that. So you don't even want to tell me then to use the second time. You must have a first. You have a first when a woman go have baby. You have a first with this and that. That no make no difference because tell me one now. If people are move forward and forward pan what do they call it now? You move forward and forward pan technology. You must have something where you can introduce to the first. So that don't even carry water, That kind of water.
Here's my goal with this video, to show you what exactly is in the new COVID vaccines that makes them work, and how they got made faster than any vaccine in history. My hope is that you'll be better informed.
But you're not a scientist, man. Ah, sir. So although you're a physician, and you know the first time, but you notice what I play. Although you're a physician, right? You're a physician. Now, you are telling me, when you hear some, you play something false or bogus? Eh? And by the way, you're still learning. Yeah? False and true or false? Eh? True or false? Because I have no physician friend, you know. True or false, ma'am? Ah, sir? Eh? Eh? Of course. Because you in your office now, and me come in there and me say me have some pain. And you diagnose me wrong. I might give me something wrong for take doctor do it a lot. You know that. Allow me to tell? Eh? 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 So, them diagnose you a lot, you know. You go in there and like them take the blood work and them do one what them diagnose and it's wrong and you're dead. I you know mash up. If you count the amount of people doctor diagnose wrong, it's unbelievable. And give wrong medication. Or you take something and then give something else. You understand? Then give something else. For hurt you more. So the doctor them kill more people than even when them come and say, well, them this and that and them is not the scientist, by the way. Still I learn.
When you get your shot, and you'll have a new appreciation for why science like this is so important. This is how to make a COVID-19 vaccine.
That answer won't move them, you know.
It turns out that some of the most important research for making the COVID-19 vaccine is happening right down the road from me at the University of Texas, which is pretty cool.
Now, if scientists took the same course where you take, yeah, you'd be a scientist by this. Me I tell you, say them study even more than that. You understand? Because you have regular family doctor, you have specialist, so you can't tell me that. You have doctor go back go study, because I have enough of them doctor there. Study more. So you don't tell me that course. That's just the normal course you just turn a doctor and then you continue study if you go do what you do. So I don't mean you tell that. If you tell me that, you tell me a lie. You understand? You take the same, you know, roll away where I turn doctor. So we take the normal thing and go through that. But you have other courses where you have each time you have to go and go take. Me now ask. So you take a look at normal doctor, if a doctor come tell me that, me as a doctor you tell me a lie now. So you take a look at doctor now and come and say, oh, me the gynecologist. Because me go study this and that. Don't tell me that. I me specialize in this and that and that and that. You know, say every doctor can go up and operate and take and cut you. You mad? Which doctor can tell me that? Every doctor tell me that name bad man. Doctor can't tell me that. None. You see me I say? No doctor can't tell me that. If the doctor tell me that, you tell me a lie. You see, you have the general doctor, them can't do nothing. They can't even get injection good. Them kill people more than anything else. See? Wrong diagnostic, but them friend cover it up. What am I talking about that? That's not joke me I make.
Because I did my PhD right here. And in here is the lab of Dr. Jason McClellan. He studies how pathogens like coronavirus cause disease.
There are 4 human coronaviruses that occur seasonally and generally cause the common cold. And then there have been 3 coronaviruses that have caused pandemics. And that's the first SARS coronavirus back in 2002, the MERS coronavirus in 2012, and now SARS-CoV-2, which emerged earlier this year.
So you notice when I play things where you can go and you can cross-check and do all these kind of things. And that me interested in it. Me not interested in talking and tell me no crap. You have some scientists, a rogue scientists them too. You know that too. You know that too, Smith. So me not fuck, I have some rogue one too, you know. Yeah, of course you have rogue scientists. So me watch them too, man. I have rogue in everything, so we know them too, man.
The end of December 2019, it was on the news that there was these pneumonia clusters in China. In the scientific community, we thought maybe a new flu virus or possibly a coronavirus. I was actually snowboarding with my family, and my collaborator Barney Graham at the Vaccine Research Center at the NIH called, and he said he's been in contact with US CDC, Chinese CDC. It looks like it's a beta coronavirus.
Of course, we know that too. We know that. Because they want to prove something and then go on and then say boom, bam, bam. And then you have something new and then something new combust on. So we know the whole of that. You see me I say? We know it. You see me I say, like when them say Venus now, them come and say Venus is not a planet. Not Venus, what them say? Pluto? Pluto them say? Not a planet? But a dwarf planet? One of them.
Similar to SARS, and they wanted to move rapidly, try and make a vaccine. And I said, we're definitely in. So you're just like scrolling through your phones in the ski lodge. You're like, we gotta get to work.
So while the rest of us were focused on royal family drama and just hearing the first mentions of coronavirus for the very first time, scientists like Jason knew this was serious, and they were already getting to work. As soon as researchers in China decoded the virus's genome and published it online, Jason's lab could start designing a vaccine.
I texted Daniel Rapp, my graduate student, and let him know to be on high alert, because as soon as we get the sequence, we're going to move as quickly as we can.
Jason was on that winter vacation and texted me that it was a coronavirus, and eventually in early January, the sequence was released online.
By the way, you have more than one coronavirus too, yeah? Yeah, because I know the coronavirus, and you have MERS, and you have SARS, and SARS are what it named before. Yeah, MERS, I think, Middle East and anything there. You have more than one of them, man.
Publicly. And then things started moving pretty quickly.
Let's step back for a minute. What does a vaccine do? Well, it trains your immune system to know what a germ, like a virus, looks like. So
the vaccine a train the system to know what the virus look like. Just listen what me I talk about. This me I talk about, you know. This is my sticking point.
Can recognize the germ, fight it off, and keep you safe without you getting sick. This is the virus that causes COVID-19. The outer shell is made of a few different kinds of proteins, but these proteins sticking off the side are the most important part, the spike. These spikes are what give this family of viruses their name, the coronaviruses, because they look kind of like a crown. The coronavirus uses that spike to sneak into our cells. The three-dimensional shape of that spike is super important because that exact shape is what lets the virus latch onto receptors on the outside of our cells, almost like picking a lock. And then it sneaks inside. Those shapes sticking out on the outside of a virus are also what your immune system is feeling for, to figure out if this is a foreign invader, if it should attack or not. The problem is, the first time your body sees a virus, your immune system responds so slowly that the virus has time to make gazillions of copies of itself. You can still get very, very sick. That's what's great about a vaccine. It trains your immune system what to look for, so when the real virus shows up, your body can respond super fast and destroy the virus before it has a chance to hijack your cells.
So your body, I move super fast to hijack yourself. So they want to train the body for your body take care of it. I will not tell you wait stand for. What that them tell me wait stand for. These are the things when make your body move slow, one know if it's a virus or not, and then it take over your cell and create one mash you up. So with this I do know is train your body. And your body is like it are the factory then. So when it that come in now, it just train it, that means say, oh, this is it, and just hold it and mash you up. Same time. You understand?
So what's actually in a vaccine? Well, sometimes a vaccine has a weakened or dead virus. That's how polio and measles and mumps and some other vaccines work. But these days, a vaccine usually just contains a little piece of the virus. The newest COVID-19 vaccines, they're just the spike.
Yeah, but if you're not telling somebody, if you train and you know that, and if you train as a physician and you work with the CDC and you know that, right? You understand. That's why I say I play this. If I play bogus thing to people, I will not do that. You understand? So we don't want to do that. I will never tell somebody say do this because we know say, you know too. The Spanish flu was starting in the US where them go give them Spanish flu over 50 million, then them start count. And it knocked them off. And we know say different virus come in all the time. You understand? So that's what me talk about. See, but you have some people just run with something and say boom bam bam and say okay, make it stay. Although we see people are dead front of our loved ones and get mashed up, and then we say boom bam bam. But like we just want no. You understand? This me want to know what in the what make the vaccine, what the vaccine do to you, and your body a fight off the thing. Wrong or right? See, everybody move slow and it can come take you over or what have you. The immune system move slow and this train your body for move even faster. For do what it do. They're not great thing that. And your body, instead of them kill the virus itself, and code it up and put now you know, them use the spike. and them I explain. So if me that tell people, I may show some people some things I tell them, well, me not another foolishness there. You understand? We want to see the world better. We need to see the world better. You see me I say? Yeah man, we need to see the world better. You understand? So that's the greatest thing. You understand? The greatest thing. That's what I want to say. So this tell you what's a vaccine. So what we want to know what's a vaccine. This I'm a sticking point what me I talk about. So if this are the sticking point, what's a vaccine? So here what me I stick about.
Copies of itself. You can still get very, very sick. That's
Someone want to get the sticking point. So what's a vaccine? So me want to know why and when you get the vaccine and wait and everything, right? You understand? So this is what me I deal with. And this is what the people them want to know. Because you want to know, when me take this, wait. Now, wait, this, what is that and that and that and that. See? So this
like a crown. The coronavirus uses that spike to sneak into our cells. The three-dimensional shape of that spike is super important because that exact shape is what lets the virus latch onto receptors.
Yes, sir, ma'am.
Almost like picking a lock.
And
then it sneaks inside. Those shapes sticking out on the outside of a virus are also what your immune system is feeling for, to figure out if this is a foreign invader, if it should attack or not.
Okay.
The problem is the first time your body sees a virus, your immune system responds so slowly that the virus has time to make gazillions of copies of itself. You can still get very, very sick. That's what's great about a vaccine. It trains your immune system what to look for, so when the real virus shows up, your body can respond super fast and destroy the virus before it has a chance to hijack your cells. So what's actually in a vaccine? Well, sometimes a vaccine has a weakened or dead virus. That's how polio and measles and mumps and some other vaccines work. But these days, a vaccine usually just contains a little piece of the virus. The newest COVID-19 vaccines, they're just the spike.
The spike alone.
For that spike to work as a vaccine,
So me I hear the spike work. So these are the things that we know, you know. Physician. You understand me? Alright, let me just call it TLLM. So this is what I want to know how it works on these things. These are them things that fascinate me. Me like know these things. You understand? So me just get up and just say, me I gotta take this and me I do this. Because we want this thing to stop. But we not gonna spread something if it's not right. We want to go through. None of them when me say I go through, them not come with these kind of things and tell me how this work, what is, this everybody sell this. Them not do that. Them just come and tell you one thing. We not in that with them. You understand? Something I forget.
Train your immune system to recognize the actual virus. It has to have the same three-dimensional shape as the spike on the whole complete virus. But making the spike all by itself, not attached to the rest of the virus, it turns out to be really hard because the spike is actually pretty floppy, just floating around on its own. It doesn't look much like the spike on the actual virus. And this is the key thing Jason's lab figured out how to make. For years, they'd studied SARS and MERS viruses, which are really closely related to the virus that causes COVID. So they already knew what tiny tweaks to make to freeze the coronavirus spikes into the perfect shape.
So these are because them that coronavirus to them, right? So them closely, so them study the SARS and the MERS. We come out from 2001 and 2012. Am I correct? Somewhere around there, some kind of work there, right, physician? And if you have that pattern, then you come and say, okay, because that's a long time. That's like 20 years ago. From the first one to the 20 years ago. So if you are continuous study as a scientist and you have built up something, that means you have this, and if this come back again, or this come back in our next thing, so make we tweak up this. Because they can tweak it. They can tweak it up. In the doctor too, you know.
So we got to work designing our stabilizing mutations into the new spike sequence. There was just two amino acids that we knew would, if we mutated them, it would stabilize the spike protein and make it a lot easier to work with in the laboratory.
A protein, like the coronavirus spike, is a long folded string of individual units called amino acids. And these strings of amino acids are built using code written in RNA and stored in DNA. By changing or mutating the letters of DNA code, we can change the amino acids in our protein string.
So it's cool. You're like building scaffolding into the protein to be like freeze in this
in this shape.
Yeah, that's a good way to put it.
How do you from there to making the actual spike protein?
I can show you.
Okay.
Scientists are able to grow special immortal human cells outside the body, which they use as factories. They put a modified gene for something like their spike protein into those cells.
And then they'll start spitting out this protein.
So they're just pumping it out into the liquid.
Yeah, that's right.
They take that liquid, run it through special purification machines, and are able to isolate a pure sample of their spike protein. But how do they know for sure that this special spike protein looks like the real thing, 3D shape and all? Well, they take pictures of it using a big, awesome science machine. This is a cryo-electron microscope. This machine took a 3D picture of the coronavirus spike and helped design the very first COVID-19 vaccine.
Check out the big science machine.
Looks like a giant microwave. I mean, the room is like $1 million, and then the microscope's another $1 million.
So you're saying don't touch this screen, right?
So you can see like the floor is separate from the instrument, so it's on its own. Vibrations are bad. These are wall panels that contain water running behind them to keep the temperature constant in the room. And then it also has to be electromagnetically shielded too.
That is nuts. This beefy cable over here.
So that's the 200,000 volts coming from that.
Okay, yeah,
so
I like that one.
Yeah.
Oh, that's just
sciencey.
Look at all that science happening in there.
Right. It's kind of a marvel of physics and engineering.
Can you play Doom on this thing?
Some of our computers, you'd be able to play at max settings.
So maybe this sounds like a super stupid question, but why can't you just use a regular light microscope to take a picture of a protein? Well, the wavelength of visible light is on the order of hundreds of nanometers. And that means the smallest things you can see with visible light are also on the scale of hundreds of nanometers. But what we want to see, the atoms in a protein molecule, they're angstroms apart, tenths of a nanometer. So we can't use visible light. We have to use a special electron microscope.
So super high energy electrons make very tiny wavelengths, which lets you see very, very small resolution things.
That's right.
Okay. I want a camera like that. That's better than 4K. We can go angstrom K.
So to take a 3D picture of a protein with a cryo-electron microscope, first you put a drop of protein onto a special metal grid. Then you freeze it in place with liquid ethane. When we shoot a beam of electrons at it, these proteins will be in all kinds of random orientations. Each orientation leaves a particular shadow. Powerful computers look at all of those 2D images and then combine them into a final 3D shape. It's kind of like using a bunch of 2D photos to make a 3D model. And when Jason and Daniel and their team looked at the spike that they made with their tiny little tweaks and mutations, their spike has the same 3D shape as the spike on the whole virus. Pretty cool. Now we can put that spike into people and see if it trains their immune system and protects them from the real virus. And it works. This protects people from COVID-19. The research you just saw from those scientists is literally what's being used in the very first COVID-19 vaccines. And some of those vaccines work in a really cool way. Instead of having to make the actual spike protein in these big factories with huge tanks of cells like the ones that we saw, some of these new vaccines, the genetic instructions for making the spike is all that's in the shot, on a molecule called the mRNA. Your body uses those instructions to make the spike. You are the factory.
You are the factory.
That's awesome. This is a...
Hold on. So them use your body as the factory now. So the mRNA. So them put it on the molecule mRNA and your body make it. Let me just play it back. So them are come with the next thing instead of them kill the thing and use like them that use.
New vaccines, the genetic instructions for making the spike is all that's in the shot, on a molecule called the mRNA. Your body uses those instructions to make the spike. You are the factory. That's awesome. This is a really incredible piece of science. A year ago, no one had ever seen this virus before. And thanks to these scientists and thousands of others around the world, now we have vaccines that work. I mean, it's going to take months, maybe years, to get these vaccines and the dozens of others still being worked on to the billions of people that need them. And that's a huge challenge on its own. But this is a really hopeful story. No vaccine in history has ever been invented this fast. And we were able to do it safely. We were able to do this so quickly because scientists like Jason and his lab and others, They were ready because they were studying basic scientific questions about other coronaviruses, SARS and MERS.
Why?
They've spent years trying
so
to...
the SARS and the MERS are like 20 years ago, right? SARS and MERS are like 20 years ago, right? Yeah? Yes. Because MERS are where? MERS are in the Middle East. When I said the Camille and them thing, right? Yeah? And then SARS about 2001. And if you have them coronavirus they study, that means you know, so okay, if something leak again, but it's you know,
figure out their secrets so that when this one showed up, they were already 10 steps ahead. And to me, that's why work like this, supporting basic research, is...
So doctor, this is what me I say to you now. So because scientists never stop work. So you go through the SARS and the MERS already, and then you have to study and you have a template, or you have to study that and study how it move again, and then the next coronavirus drop. They might move to that because they might have something. Because you know, when it's done and you clean up SARS or clean up MERS, I say, okay, I'm done. I'm a dash for everything. I'm a stop study about it now or something. No, not even smallpox, because that thing them still have it in a lab, you know. Although them say them eradicate it, right? But no, them eradicate it, yes. But them still have it in a lab somewhere there. Because that's how the country them running now. Yeah, they eradicate something, but them still keep a sample or something there. That if it let out a lab, you know, because little later can turn a biological agent or something. We shouldn't have said it, but a reality. See? So you have to study how the world runs. So if something happened and them have had a coronavirus before, see? Them still have to study after that. I know so something can tweak up and start do something else because this come already and the one they come to. So they know, so okay, so I don't know them must study this. I don't know. So we're going to look on, and next time we're going to look on SARS and MERS, you know, and see, and look on them damage and how them control and do them things, you know. You see me I say? Because, yeah, we have to have explanation on certain thing, you know. You see me I say? You see me I say? Yeah, man. Me like, go in and see. Me not talk when people are sending off where them are sending. I want to know, you know, how antibiotic make, where it do, where this do, where the vaccine do, how they make it, how it design, where your body, because them put less of the germs itself for them to have in a year and then run with it, you know. So these are the things that me like ask. See, these are the things that I want to watch. Me not say nothing over now. Nobody not gonna see me I post up some things where, you know, unnecessary. When me look, this thing reach at my house. It reach at my house. Thank God we never have the bed. But it reach at my house, so I have to say, so it draw me down more to some things. You see me as a doctor? You understand? It draw me down more and say, you have to put a eagle eye pan a thing. You understand? It have to move. SARS and MERS, them tear me too. Because it did go on bad and them hold it down. And they keep 100 days of pandemic. Because you have it before. You have the Black Plague. You have all kind of little things run before. You see what I say? Nothing. You have the Spanish flu. When them reach 50 million, them estimate 50 to 100 million. In the 1918 to 1920. Just to over that. 2 years. 50 million them say, but look upon it estimated. 50 to 100 million. Because like them lose count, and that's enough people for lose back in the days. Polio come again and do a lot of damage. Nothing come and do a lot of damage. But thank God for these scientists and them think that them come and pull off something, you know. You understand? And then technology more increase right now. Remember, look how technology increase. One time just in the 70s, that's when I'm not talking in the 80s, because 70s TV gen and broadcast, them carry them little thing on black and white TV. But in the 80s when they want to do a live broadcast outside, them have to carry this big old van and some other things and bigger and push up something and some big old satellite dish we have and them think they, you know see people them big old 25 feet satellite dish across and 30 and 40 and 50 feet, big like your stop. You don't see them thing there again. I will make a call and find out, I go jam in tell. And pay money and set up my line. I will say a call back. We don't see these things again. Because things, you understand, technology increases, you know, man. The phone, when we just get all about phone, we couldn't even do them things with it just the other day. You see me I say? We couldn't do the, you see me I say? Yeah, man, doctor, you're up, doc. You're up. But we are man, we like see them thing and we want to see the whole world, you know. So we now discourage things when we say life a loss and them thing there. I mean I say boom bam bam just me say cross reference and do the thing and do the research. And just take where man I say. Just go and want to know how them come towards this or see if it is and everything. Just do your thing and make sure the thing up. You see me I say? Yeah man. We say the policeman break the law. Well, them break laws all the time, you know. You see me I say them break laws all the time, so then I go, them break the laws all the time, man. I say it go man. We want to hold it up, you know. Me there, you know, keep it real. Yeah man, everybody big up on yourself every time, yes sir. Big up, yeah? Yeah man, Lonesome, keep it real, yeah? Well, up.